Hello, I'm folate, a vitamin. I'm also known as vitamin B9. Vitamins are coenzymes that support chemical reactions happening in your body. I get activated in the liver, and that's when I start doing my job. Let me explain in a bit more detail. I'm made up of 3 parts: a pterin ring, benzoic acid, and glutamic acid. Among these, the pterin ring is originally stiff and not very reactive. But when 2 hydrogens are added to it, it softens up and becomes an active form that can participate in chemical reactions in the body. That's when I can start carrying what's called a 1-carbon unit. 1-carbon units are functional groups that contain just 1 carbon atom. For example, methyl CH3, methylene CH2, and 4-mil CHO groups. These 1-carbon units attach to me at 2 specific nitrogen positions, N5 and N10. Methyl group attaches to N5. 4-mil group attaches to N10. Methylene group bridges between N5 and N10. Today, I'll explain how I help recycle homocysteine into methionine by carrying a methyl group. Cysteine, homocysteine, methionine, SAM. This cycle plays 2 important roles in the body. First, it regulates genetic switches through methylation. Second, it creates antioxidants that protect your cells. So, what exactly happens when genes get methylated? Let's start with the cytosine bases in the promoter regions of DNA. When these cytosines get methylated, the gene becomes harder to read, and its expression turns off. Also, DNA is wrapped around proteins called histones. When these histones are methylated, the tightness of the wrapping changes, and that affects how easily the gene is read. By the way, histone methylation can either turn genes on or off, depending on where and how the methyl group is attached. And who delivers this crucial methyl group? It's me, folate. Alright, time to deliver my methyl group to homocysteine. Wait, I'm a little shy, so I can't hand it over directly. Instead, I ask my friend vitamin B12 to pass it on for me. From vitamin B12 to homocysteine, methionine is formed. Methionine then picks up ADP to become S-adenosylmethionine. Let's take a look at SAM's structure. It has a methyl group from folate and adenosine from ADP, and the base is homocysteine before it got modified. The sulfur holding the methyl group is very unstable. It desperately wants to donate the methyl group to someone. And its targets? DNA, histones, and other genetic switch parts. After giving away its methyl group, SAM turns into S-adenosylhocysteine. Then, once adenosine is removed, it goes back to homocysteine. When a CH2 group is added to cysteine, it becomes homocysteine. Then it receives a methyl group and an adenosine from ADP. Now it becomes the high-energy molecule S-adenosylmethionine. Once SAM donates its methyl group to DNA or other molecules, its job is done, leaving behind homocysteine and adenosine. The homocysteine can be recycled. But too much homocysteine can build up and cause arteriosclerosis. So your body does 2 things to manage it. Recycles it back to methionine with help from folate and vitamin B12. Converts it into cysteine using vitamin B6. Then uses that cysteine to make glutathione. Next time, we'll tackle a must-know exam topic. It's full of similar-sounding names and super confusing. Every like and subscribe gives me the energy to make the next video. See you in the next video.
For quick, powerful reviews, this is the only channel. Don't forget to subscribe.